I am super thrilled to be speaking to all of you today. I am a fourth year graduate student in Dr. Ami Bhatt's lab at Stanford, and I'll be talking to you all today about a really exciting collaboration to study the gut microbiomes of South African women. So when we compare populations from around the globe, we've seen that communities from different geographies have very different gut microbiome compositions. And we know that the composition of the gut microbiome is really intimately connected to changes in diet, in lifestyle, in resource access, et cetera. And we've seen this illustrated in lots of different publications recently. One in particular I'm showing here, Almeida et al. In 2020, in this very large effort to catalog the full range of microbial diversity, they built the Unified Human Gastrointestinal Genome Collection, which did a really beautiful job of compiling the full set of known gut-associated bacterial genomes from around the world. And this visual here from Almeida et al. Summarizes the number of gut-associated microbial genomes generated from each country. And as you can see in red, you have the US, you have China, you have parts of Europe that are really highly represented in microbiome research. And conversely, you have a really small handful of microbiome studies looking at communities at the other poles of resource access. So you have, for example, you see hunter-gatherer communities represented from Tanzania, agriculturalist communities represented from Madagascar, but the vast majority of low and middle income countries, which happen to hold half or more of the world's population, are almost entirely excluded from microbiome research. So our group had the opportunity to begin a collaboration with a team at the University of the Witwatersrand in Johannesburg, South Africa, and we sought to study the microbiomes of adult South African women. And this collaboration was made possible by the H3 Africa Consortium, which is a continent-wide consortium that's involved in a huge range of projects, largely studying genetic and environmental factors that influence disease. And our study in particular was a subset of the AWIGEN project, which is an H3 Africa project studying risk factors specifically for cardiometabolic disease across 4 African countries. So we were able to leverage existing study sites and participants from the AWIGEN project, and we focused specifically on 2 South African communities, one being urban Soweto in the top left, and the other being rural Bushbuckridge in the top right. So Soweto is an urban township. It borders Johannesburg. Bushbuckridge is a former apartheid homeland in northeastern South Africa. And as you can see from the table here down below, Soweto is much, much more dense, much more urbanized, and has an increased proportion of households with access to piped water and flush toilets. And so from these 2 communities, we assembled a cohort of just under 200 adult women, and we collected single stool samples from each individual. And those samples were then profiled with 16S and then shotgun metagenomic sequencing. Our collaborator at WITS, Foke Udwaran, who's pictured down here below, recently published the 16S work. I highly suggest that you read it. It's an excellent piece. But today I'll be focusing on our shotgun sequencing and long read sequencing work. So from our shotgun data, we found that the microbiomes from individuals in rural Bushbuckridge had much higher alpha diversity than their Soweto counterparts. And this is something that we might have expected beforehand because prior works have shown that there is a decrease in gut diversity with increased industrialization. We also identified a number of genera that were significantly enriched in one community over the other. For example, you see in Bushbuckridge that there's an enrichment for Sucinetamonas. That's one of those hallmark taxa that's lost with industrialization. Conversely, you see, for example, that the Soweto microbiomes are enriched for taxa like Bifidobacterium, Bacteroides, some of those genera that have been shown to increase with industrialization and urbanization. Something that I actually thought was particularly interesting, though, is that we see that microbiomes from both Bushbuckridge and from Soweto had a non-zero abundance of certain taxa that have been described in the literature to vanish with industrialization. So we see, for example, that taxa like spirochetes, treponemas pictured here in the middle, are actually present even in these industrial urban communities like Soweto. And that actually goes quite against what we as the field had thought previously by exclusively focusing on very, very rural communities like the Hadza. And so, having observed all of this, we really sought to contextualize some of these observations with those of communities at those more extreme ends of diet and lifestyle. So, we accessed publicly available shotgun sequencing data for adult industrialized cohorts in the US and Sweden, pictured here in blue. And then we also accessed publicly available data for cohorts from rural Madagascar, from the Hadza in Tanzania, and from a recently published cohort in Burkina Faso. And so at a very high level, when we compositionally compare these metagenomes, we find that the South African microbiomes are somewhat intermediate between the Swedish and American cohorts, again in blue, and then the Tanzanian, the Malagasy, and the Burkina Faso cohorts in shades of red and orange. But what I find kind of interesting here is that the South African cohorts here in purple don't just cluster on this MDS in a very simple linear continuum across that industrialization axis. And in fact, they seem to occupy a unique position in this multidimensional space. But we realized that this sort of analysis probably wasn't telling us the whole story, and sought to obtain a measure of how much unclassified microbial diversity might be present in these cohorts. And so we took all of our shotgun reads and looked at what proportion of those reads were able to classify to reference databases of known microbial genomes. And we found, as you can see here, that the less industrialized communities, so Tanzania, Madagascar, Burkina Faso, and the two South African communities, had a much lower proportion of their shotgun reads that classify to reference databases. And that led us to ask the question of which taxa account for these unclassifiable reads. So what taxa are present in these microbiomes that are absent from reference databases? And so to address this question, we decided to perform de novo genome assembly on the metagenomic reads. So assembly of microbial genomes from metagenomic sequencing follows a really similar process to assembly of any genome out there, but you have the added complication of having to disentangle different taxa in your sample from one another. So the basic workflow is that you extract DNA, you sequence it, you assemble your reads into contiguous sequences or contigs. Those contigs are then grouped together into draft genomes or bins based off of different features. And each bin then represents a single microbial genome. But as you can see from my schematic, you do need to proceed with caution in a case like this because assembly and binning can be imperfect processes. But when we went through this assembly and binning process for all of the participants in our study, we were able to assemble thousands of medium and high quality microbial genomes from a wide range of bacterial taxa. So here I'm showing all of the genomes from all of the samples in that inner phylogenetic tree. The inner circle shows the phylum of every genome. The middle circle shows you the site that every genome belonged to, so what participant corresponds to that genome. And then the outer circle shows you the novelty of every genome compared to a reference database. So you see in some of those bright green stripes around that outer circle that we actually identified novel mags corresponding to new strains, new species, new genera, and beyond, even from a study that was relatively small compared to the hundreds of thousands of reference genomes we were comparing to. And then you'll also note that there are some genomes in that inner tree that are marked with a big green circle, and those genomes were generated with nanopore sequencing. So we had decided to perform nanopore sequencing on just a very small handful of the South African samples because we wanted to give ourselves the best shot to generate really complete bacterial genomes. And so we were able to assemble highly contiguous genomes for taxa that have no public references available. So here I'm representing contigs as blocks of color along a circular axis. The nanopore assemblies are in green, the short read assemblies are in gray, and then you have inner tracks in pink and yellow representing the GC content and GC skew of each genome. And you see that we assembled really complete genomes for taxa like B. vulgatus, Prevotella, those really well-known taxa that lack good references because their genomes contain so many mobile elements and repetitive structures. We also have single contig Treponema genome. So Treponema is one of those taxa that's lost with industrialization, very hard to culture in a lab. And then we also have examples of complete genomes for taxa like bacteria, which has very low GC content and can't assemble well from short read data because of GC bias in those approaches. And these genomes tell us a lot of important information because complete genomes can show us more than fragmented shotgun genomes. So to illustrate this, we identified and annotated regions in all of our nanopore genomes, shown here again in green, that weren't covered by those gray shotgun contigs. And as you can see in this cartoon, we identify elements like those in blue that weren't present in the shotgun data, but are present in the nanopore genomes. And when we annotated those new elements, we see that they're enriched for regions of horizontal gene transfer or possible mobile elements, transposases, recombinases, phages, antibiotic resistance genes. And those elements can have really important effects on bacterial phenotypes. And this really highlights the importance of using techniques like long read sequencing to move beyond just classifying taxa and towards really understanding the functional components of a metagenome. So with that, I'll conclude by saying that we performed the largest gut microbiome shotgun sequencing project with adults in Africa and found that these microbiomes really contain a lot of undescribed novelty. And we push against the dogma that all of these intermediate populations exist on a simple spectrum. And we are also able to uncover novel genomes using advanced metagenomic techniques. And this really shows us that we can explore the vast diversity of the microbiomes of understudied communities using methods such as this. I'll close by thanking the many, many collaborators involved in this work. Largest acknowledgement to Fiona Tamburini, who truly was the lead student on this work for many, many years. And I'd also like to thank our collaborators at WITS, at the study sites, and the participants in the study. With that, thank you for your attention. I'm looking forward to the Q&A.